This will continue with the metabolism lecture. This will be part 2. So we're just going to start the aerobic carbohydrate metabolism section, which includes glycolysis, pyruvate activation, Krebs cycle, and then the electron transport chain. So the first step in the aerobic metabolism of the carbohydrate is glycolysis again. Okay. Now, in the previous video, we looked at anaerobic glycolysis, which started with glucose and actually ended with lactic acid. And that's because we converted the pyruvate that we made into lactic acid in the anaerobic conditions. In the aerobic conditions, we're not going to do that, but we're going to take the pyruvic acid and move it into the mitochondria. So in many ways, this is similar as to what we saw before. We still have to put 2 ATP in. We get 2 NADHs that we make. We get 2 ATP for each side, so 4 ATP that we make. But unlike the anaerobic glycolysis, where we had to use this NADH up to make lactate, we get to keep this. Now you're going to hate these. You'll see why in a few minutes. So we get to keep the NADHs and eventually cash those in for energy in the mitochondria, because this is the aerobic metabolism one. So once we get through glycolysis, we're going to take our pyruvate, and pyruvate gets transported into the mitochondria. And we're going to convert pyruvate into something called acetyl coenzyme A. Pyruvate is a 3-carbon molecule. Acetyl coenzyme A, you can see the carbons in red, is 2. So 3 carbons, this carbon, these 2 red ones, and inside we remove the carbon and the 2 oxygens, CO2, and replace it with a coenzyme A. So that's why we call this 2-carbon unit acetyl, and since there's a coenzyme unit, we call it acetyl coenzyme A. So what we've done is we've broken a large bond in the activation step, got rid of a CO2, so we produced a CO2 in the mitochondria, and made an NADH in the process of making acetyl coenzyme A. So that's the activation step. Now, why is it 2? Well, remember, for 1 glucose molecule through glycolysis, we got 2 pyruvate, which means we're going to get 2 acetyl coenzyme A's, 2 CO2s, and 2 NADHs. Okay. Now, these NADHs are a little easier to deal with, and again, we'll look at that problem in a minute. So for the Krebs cycle, right, this also occurs in the mitochondria. Remember, we're inside the mitochondria now, so we don't have to worry about it. We're going to take 2 acetyl coenzyme A, so we're going to go back up here. What we're going to do is we're going to combine these 2 acetyl coenzyme A's, we're just going to do it one at a time though, with a molecule of citrate. Now, citrate, remember we said it's the same thing as citric acid. Remember, the ic acid is the same as the 8. So we take this 2-carbon acetyl coenzyme A and oxaloacetate, which is 4 carbons, and we form the citrate molecule, or citric acid. So that's why this is called the citric acid cycle, because the first intermediate we form is called citric acid. Okay? And, let's kind of look at the carbons again. I have a 4-carbon, this is called oxaloacetate, or OAA, and a 2-carbon, acetyl coenzyme A, and that forms a 6-carbon, so 1, 2, 3, 4, 5, and the 6th one out here on the side, of citric acid. Okay? And then you don't have to worry about these steps. Go through a series of steps. Series of steps. And eventually you end up at oxaloacetate again. So that's why it's called a cycle. So as long as I'm putting acetyl coenzyme A units in it, I can go around this circle and keep on doing oxaloacetate. Okay? So what happens in this process? Okay, so this is called one turn of the Krebs cycle, like one little wheel. Okay, so in one turn of the Krebs cycle, let's kind of look at what we get, and they've done a pretty good job of kind of summarizing things. So if you look right here in purple, you get a CO2 and a CO2. That makes sense, again, because our first step, we have 6 carbons. Our last step, we have 4 carbons. What happened to the other 2 carbons? They came off as CO2. So we get 2 CO2s for each turn of the Krebs cycle. For each acetyl coenzyme A we stick in, we get 2 CO2s. We get 1 ATP. So if you look down here, you get ATP. So that's substrate level phosphorylation. That's direct ATP. We get 3 NADHs. So see the arrows coming out? 1 NADH, 2 NADH, 3 NADHs. And we get 1 FADH2 here. Okay? And notice all these arrows are pointing to oxidative phosphorylation. Okay? That happens in the electron transport chain where we turn these high energy nucleotides, that's what they are, nucleotides, energy carriers, into ATP. All right? Now, again, one turn of the Krebs cycle gives me 1 ATP, 3 NADHs, 1 FADH2, and 2 CO2s. But for one glucose molecule, since I have 2 pyruvates that both became activated, right, and then I have 2 acetyl coenzyme A's, I get to do this twice. So that's why over here we have 4 CO2s, 2 for each turn of the Krebs cycle, 6 NADH, 3 for each turn of the Krebs cycle, 2 FADH2, 1 for each turn of the Krebs cycle, and 2 ATP, 1 for each turn of the Krebs cycle. Okay? So again, to go back to some terminology, how many ATP do we get through substrate level phosphorylation for one turn of the Krebs cycle? Where you look here and you go, okay, one turn is this. How many direct ATP do I get? 1. How many substrate level phosphorylation ATP do I get? 1. It's the same question. Right? If I said indirect, it's a little more difficult, but we can do it. So I have 3 NADHs, right? Remember, each NADH is worth 3 ATP. So that's 3, 6, 9 indirect through oxidative phosphorylation. And then that FADH2, remember we said that was worth 2. So let's go ahead and do it this way. All right, so I'm just going to make them pink because it's easy to see. So we have 3 NADH, we get 3 ATP from that. We get 3 ATP from that. We get 3 ATP indirect from that. And this FADH2 is worth 2. So 3, 6, 9, 11. So for one turn of the Krebs cycle, my indirect ATP is 11. Another way to say it is for one turn of the Krebs cycle, the ATP I get from oxidative phosphorylation, that's the same as indirect, right, is 11. Now, if I want to count my direct ATP, I'll come over here. That's the only one I have, so that's 1. So my direct ATP, or substrate level phosphorylation ATP, is 1. Well, what about my CO2? Well, we saw the CO2 before. I get 1 here and 1 here. So I get 1 CO2 here and 1 CO2 here. So that total is 2 CO2 net that we lose here. Okay. So those are some more of the important ones. I guess we could do one more, the total ATP. The total ATP is your direct plus indirect. So my indirect again was 3, 6, 9, 11. My direct is 1, so that's 12. So my total ATP is 12. What about my net ATP? Remember, net ATP was the total minus the used. We just said the total was 12. I didn't use any because we're just talking about this one turn of the Krebs cycle. There's no ATP we have to use here. So it's 12 minus 0. So the total, 12 minus used, 0. So the net ATP is 12 as well. As a matter of fact, the only place that we use ATP as far as we're concerned in terms of the metabolic processes we're going to study to be responsible for is at the beginning of glycolysis. That's the only place where we use ATP. So let's kind of look at the electron transport chain then. We're not going to worry too much about the electron transport chain. It's a little complicated and there's not a whole lot I want you to remember from it. But basically, the things I do want you to remember is just like the activation of pyruvate, just like the citric acid cycle, the electron transport chain, oxidative phosphorylation occurs in the mitochondria. And basically, it's the place where we can turn NADH and FADH2 into usable energy. Okay, so it's not a bad analogy would be like a casino where you used chips, right? So if you've ever been to Las Vegas or other casinos where you have chips and then you turn the chips in for cash, right? That's basically what the electron transport chain is. It's like the cashier's cage, okay? And so we want to be able to use the NADH and FADH2 as energy, but to do that, we have to convert them to ATP first, and that happens here in the electron transport chain. Okay? And that process is called oxidative phosphorylation. So don't worry too much about the equations. That's true for all of them. You should worry about the summary tables, certainly, but not too much about the equations. And so let's kind of just explain what the electron transport chain is. It's a series of enzymes located in the mitochondria, okay, where what happens is we pump hydrogens across the membrane and then let them diffuse the other way across the membrane and synthesize ATP. Okay, it's a pretty simple process from that standpoint. And that whole process together, and this is the key, is called chemiosmosis. So the chemiosmotic theory explains how that works. So basically it involves pumping hydrogens across the mitochondrial membrane and then letting them diffuse across so we can make ATP. Okay? Now, electron transport chain is just a series of proteins. They're enzymes, basically. And the reason why NADH gives us 3 ATP and FADH2 only gives me 2 ATP is where they enter. NADH enters at the beginning of the pathway. So if you look right here, here's NADH entering the first cytochrome, the first protein, basically. And so that allows me to move one hydrogen across the mitochondrial membrane, which then is going to result in making an ATP. FADH2 enters later, and so it enters after this first step, so it can only make 2 hydrogen ions move across, which make 2 ATP, but NADH does the first one, then it does the next 2 as well, and so that's why we get 3 ATP. And it really has to do with specificity, right? And NADH, fits into this first protein, and FADH2 doesn't fit until later on. It doesn't have the right 3-dimensional shape. Okay, I want you to know that it's hydrogen ions that are pumped and then diffuse across that help make ATP. I want you to know that's called the chemiosmotic theory. I want you to know the energy equivalence. NADH gives me 3 ATP. FADH2 gives me 2 ATP. I want you to know that this happens in the mitochondria and oxygen is required, and that the process of making NADH and FADH2 into ATP in the electron transport chain is called oxidative phosphorylation or indirect ATP. It's a nice summary of what we're doing. Now, here's one of the problems that we have, and that's when we're going to look at the economics of what's happening. This all occurs in the mitochondrial membrane, okay? And that means that in order to have NADH or FADH2 made into ATP, I have to get the NADH or FADH2 into the mitochondria if it happens outside. Well, the good news is the activation of pyruvate, where we make 2 NADHs, one for each pyruvate, that happens in the mitochondria. Citric acid cycle happens in the mitochondria. So the 3 NADH we make there and the 1 FADH2 we make there for one turn of the Krebs cycle, right? That happens in the mitochondria. But in glycolysis, that happens in the cytosol. And again, let's go back to our organelles. Mitochondria is a membranous organelle. It has a cell membrane. And in order to move across the cell membrane, you have to have 2 properties. You need to be small and nonpolar. So you're seeing how, we're building upon some of the stuff that we learned last time. And if you take a look at the NADH molecule, it's a nucleotide. And again, going back to the biomolecules, all nucleotides have a couple things. They have a sugar, right, which is polar. They have a nitrogenous base, which is polar. They have a phosphate, which is polar. So nucleotides tend to be very polar. So NADH, which is a nucleotide, cannot pass through the mitochondrial membrane. So how does it get its energy into the mitochondria to make ATP out of it? Well, there are 2 basic ways to do it. Okay, so you should know both of these mitochondrial shuttles. One happens primarily in the heart and liver. As a matter of fact, in every cell in the body for the most part, except for skeletal muscle, this happens. It's called the malate aspartate shuttle. Okay, and basically, you don't have to worry about the details, but the electrons from NADH, the energy, are passed to a molecule called malate that can diffuse across the mitochondrial membrane and pass that energy back to an NADH molecule inside the mitochondria, and then it diffuses back out as aspartate, and it just keeps on going back and forth. So it's called the malate aspartate shuttle. It is a passive transport mechanism. Okay, and so there is no energy required. There's no energy to input, and it just happens based on the concentration differences. And really, you look at it, how much energy is outside in the cytosol versus how much energy is inside the mitochondria. Heart and liver typically don't have a huge requirement for ATP. The heart maybe a little more, but the liver not much. The liver needs ATP to do its cellular processes, but it doesn't need a lot of ATP very quickly like a skeletal muscle would. So it can wait for the passive transport to have the concentration gradient to diffuse into the cell to make ATP. Skeletal muscle, on the other hand, especially during high levels of exercise, it needs to make as much energy as possible. So it uses a different system, and that system, for the most part, think of it as an active transport system. And in an active transport system, you're going to have to put energy in. And so what happens in this, and this is a little more complicated than it needs to be, but basically the energy from NADH is passed to a molecule called glycerol phosphate. And think of it as an active transport mechanism where even though we had NADH on the outside, we have to use an ATP to get that molecule on the inside. Think of it as paying a toll. right? So if you had a toll bridge where you had to cross and it cost you $1, and you had $3 before you went over the bridge, spent the dollar on the bridge, and then came across, you'd have $2 left. And so basically that's what happens here in the glycerol phosphate shuttle. The energy from NADH, right, is passed. The glycerol phosphate moves across. It costs 1 energy unit. So by the time it's inside, it doesn't have the same energy it had when it was outside because it's lost in a phosphate. It's lost in ATP basically. So now it only has enough energy to pass to an FADH2, which means in this case, I only have 2 ATP. And you might think, well, why would you do that? Why would you have a system where you lost an ATP molecule? Well, here's the positive part of it. Because it's in skeletal muscle, and skeletal muscle has a high need for oxygen, for ATP at certain times when it's under high contraction load, it's willing to give up 1 ATP to be able to actively transport that energy into the mitochondria to make right now. And it's similar to, those, little payday loan things, right? Every quarter, it seems like, where let's say, your car broke down and, it's still a week left in the month and you don't get paid for a week, but you can't get to work because you can't fix your car because you don't have the money. So you go one of those short-term loan places, borrow $500, get your car fixed so you can go to work and make money, but have to pay them back $600. And you're willing to pay that little extra to get that money now. And the skeletal muscle is the same way. It's willing to pay 1 energy unit, basically 1 ATP, to be able to get that energy made into ATP right now, instead of waiting for a concentration gradient where the cell may not need that energy and it doesn't really care then if you make it as much. So that's the glycerol phosphate shuttle that occurs in skeletal muscle. So in heart and liver, we use the malate aspartate shuttle, and we get, for every NADH we have, we get an NADH inside the mitochondria. In skeletal muscle, we use the glycerol phosphate shuttle, and for every NADH we have outside the mitochondria, we only make FADH2 inside, so we only get 2 ATP per NADH molecule instead of 3 like we do in the heart and liver using the malate aspartate shuttle. So this is not a bad graph, although it's not completely correct. This shows some of the things that can happen. So we have glucose, we make some pyruvate, right? Pyruvate goes into the mitochondria, make acetyl coenzyme A, and then here, right, we make 2 ATP, we get our 4 CO2 out. So this is 2 turns of the Krebs cycle, right? And then we get 2 NADH that cross the mitochondrial. This is where we get our 2 NADH from the activation of pyruvate. This is where we get our 6 NADH from the Krebs cycle, 3 for each turn. So we got my 2 FADH2, each one for 1 turn of the Krebs cycle. And that's where we cash it in for energy. So this is where, right, it says 32 to 34 ATP. The reason why there's a range, okay, so the reason why there's a range is because it depends on what shuttle you use for this NADH. Okay, so that's why it's there. Now, why does it say 32 to 34? Is because this is from a book that uses the actual yield of ATP instead of the theoretical yield. Remember, we're saying we're using the gross or theoretical yield, where 1 NADH gives me 3 ATP, 1 FADH2 gives me 2 ATP, while this book is using the actual yield where it's saying, okay, I only get 2.5 ATP from an NADH and 1.5 ATP from an FADH2. So it makes the metabolic economics a little differently. And I think there's a better way to kind of summarize this, and that's with this table. So we saw a similar table for anaerobic glycolysis. And so we're going to look at this one for the whole complete oxidation of glucose, which means breaking it down completely. When we do that, we have to specify where we're at and what shuttle we're using. So the first one will be the different steps in terms of breaking down glucose aerobically. And we're going to do this one, as you can see here, in the heart and liver. Okay, so the first process is glycolysis. We always use 2 to get glycolysis started. We get 4 ATP direct. Remember, this is for a whole glucose molecule. We get 2 NADH in that process. This is NADH net. And remember, using the heart and liver and the malate aspartate shuttle, the NADH that we have outside the mitochondria gets transferred to NADH inside the mitochondria. So that works. All right, so that's 2. No FADH2 here yet. ATP indirect. This is how much ATP we get from NADH and FADH2. So again, for NADH, you should times it by 3. And for FADH2, you should times it by 2. So 2 times 3 is 6. 2 times 0 is 0. So 6 plus 0 is 6. So my ATP indirect, this 6, comes from the FADH2 net and the NADH net. Okay. My total ATP is 10. How do I get 10? I take the indirect, 6, plus my direct, 4. So 6 plus 4 is 10. What about my ATP net? My net ATP is total minus U. So I have 10 total, 2 U's, so that's 8. And remember, no CO2 came off at this time. Remember, we start with a 6-carbon glucose and make 2 3-carbon pyruvates. So I still have 6 carbons, so there's no CO2 produced. Let's look at the next step. Now, glycolysis in skeletal muscle is going to be different. And the difference we've talked about already, but let me remind you, is even though we make NADH, we always produce NADH in glycolysis. This NADH in glycolysis in the liver and heart, using that malate aspartate shuttle, we still made an NADH inside the mitochondria, didn't worry about it. But in skeletal muscle, because of that active transport mechanism, it's going to cost some energy to get it into the mitochondria. So we don't have enough energy in NADH to pass it back to NADH, so we have to pass it to FADH2. So these are net. So even though we made an NADH, we don't have one anymore. And what we did is we replaced it with an FADH2 in the mitochondria. Okay, so the cytosolic NADH, that means the NADH produced in glycolysis in the cytosol, gets converted to FADH2 in skeletal muscle. And once you do that, everything else is the same. So FADH2 net, we have 2, right? And so that's where the difference is. But everything else is the same. So we'll go start at the beginning. ATP used is 2. We always put 2 ATP in. ATP direct, we always get 4 ATP out. 2 for the top one, 2 for the bottom one. NADH net, there's 0 because we converted to FADH2 because we lost 1 ATP's worth of energy moving across the mitochondria, right? So we have 2 FADH2s. Remember, this is times 2, so that's 4. This is times 3. 0 times 3 is 0. 0 plus 4. My indirect is 4. My total ATP is indirect 4 plus direct 4. That gives me 8. My net ATP is 8 minus 2 used. That gives me 6. Still no CO2 out, right? So that's glycolysis in skeletal muscle, okay? When you have the complete oxidation of glucose with aerobic pathways, breaking glucose down completely and using it for energy. Now, pyruvate activation happens the same no matter what. So whether it was the heart or liver or skeletal muscle, it doesn't matter. Once we get into the mitochondria, everything else is the same. So the pyruvate activation, okay, we turn 2-pyruvate into 2-acetyl coenzyme A's. In that step, we get 2 CO2s and 2 NADHs. So going back to the beginning, for the activation step, 0 used, 0 direct, we didn't make any ATP out. We have 2 NADHs, 1 for each pyruvate we activated from that glucose molecule, 0 FADH2. So indirect, 2 times 3 is 6, 0, 0, 6 plus 0 is 6. Total, indirect plus direct, 6 plus 0 is 6. ATP net, total minus used, 6 minus 0 is 6. CO2 net, for each pyruvate we activate to acetyl coenzyme A, we get off of CO2. Since this is for a whole glucose molecule, we get 2 pyruvates per glucose, so it gives us 2 CO2 nets. And again, this happens the same in the liver or skeletal muscle. Krebs cycle is the next step. Again, this is also known as the citric acid cycle, or sometimes even the tricarboxylic acid cycle. Sometimes people even call it TCA for short, for tricarboxylic acid. When I was an undergraduate student, I did not know that. And I had a professor that said, oh, we're going to learn about TCA today. And started talking about it and was looking at it and go, wow, it looks a lot like Krebs cycle. And it was. So I just didn't know it was called that as well. So there's got like 3 or 4 names. So we're going to call it Krebs cycle, though, even though it's an eponym. I think most people understand it as that. So for the Krebs cycle, we don't have to use any ATP. Again, the only time we use ATP is at the beginning of glycolysis. ATP direct, remember 1 turn of the Krebs cycle gave us 1 ATP. And since we have 2 turns per acetyl coenzyme per glucose, because we get 2 acetyl coenzyme A, we get 2 direct. My NADH net is 6. Remember, 1 turn gives me 3. The other turn gives me 3, so that's 6. My FADH2 net, I get 1 per each turn, so that's 1 plus 1, that's 2. Indirect ATP is 22. How do we get there? Well, NADH net is 6. 6 times 3 is 18. FADH2 is 2. 2 times 2 is 4. So 18 plus 4 is 22. My ATP total, indirect 22 plus direct 2 gives me 24. My ATP net, total minus used, 24 minus 0, 24. All right, so my net is 24. My CO2 net is 4. Remember, each turn of the Krebs cycle gives me 2 CO2s. We take a 4-carbon oxaloacetate, a 2-carbon acetyl coenzyme A, make a 6-carbon citric acid in the citric acid cycle or Krebs cycle. It goes through the various steps and ends back in an oxaloacetate that's got 4 carbons. So we lose 2 carbons each turn of the Krebs cycle, and those come off as CO2. So this is the total when we stick all these together in one table. ATP used is 2. ATP direct is 6. Since we're in the heart and liver using the malate aspartate shovel, we're going to keep the NADH. So the total NADH total is 10. 2 plus 2 plus 6 is 10. FADH2 net is 2. My ATP indirect, there's 2 ways to figure it out. 6 plus 6 is 12, plus 22 is 34. The other way to check it is go, okay, NADH, I have 10. 10 times 3 is 30. FADH2, I have 2. 2 times 2 is 4. 30 plus 4 is 34. ATP total, 10 plus 6 plus 24 is 40. Or 34 plus 6, direct plus indirect, is 40. ATP net, 8 plus 6 plus 24 is 38, or 40 minus 2 is 38. And the CO2 net is 2 in activation, 4 in Krebs for the glucose molecule, and that gives us 6. Remember also that if we're completely breaking a glucose molecule apart and liberating its carbons as CO2, and we start with a 6-carbon sugar, we're going to get a yield of 6 CO2s. Now I'm going to show you the skeletal muscle one, right? And the only difference, we go back and forth, the only difference is right here. This NADH net now becomes FADH2 net because even though we made these, we had to use some energy to transport it across the mitochondrial membrane in that active transport mechanism. So this becomes 2 now. But everything else is the same except for the math that makes this different. So we still use 2 with glycolysis used. We still have 4 direct in glycolysis and 2 direct in Krebs, so 6 direct. But the NADH, since we don't have it in glycolysis anymore because we converted to FADH2 instead, we have 2 plus 6 is 8. FADH2 net is 2 plus 2 is 4. So how many ATP indirects did I get? Well, 8 times 3 is 24. 4 times 2 is 8. 8 plus 24 is 32. And you can add these up. 6 and 4 and 22 is 32. And then you could add the direct. 32 plus 6 is 38. You can add up this column as well, which will be 38. ATP net, you can subtract the 2 out and get 36, or add up this column, you get 6 and 6 is 12, plus 24 is 36. And then our CO2 net is the same. We still get 2 in activation, 1 for each pyruvate activated acetyl coenzyme A, and then 4 total from Krebs cycle because we get 2 per turn when we turn that 6-carbon citric acid into a 4-carbon oxaloacetate. So carbohydrate catabolism means to break down carbohydrate. It's only about 40% efficient, right? And when we have inefficiency, that rest of that energy comes off as heat. All right, you don't have to worry about this, but just trying to show you in the steps as we break down and get ATP basically out of this glucose molecule. Well, if you look, and this kind of makes that point I said about small and big bonds, notice this drop here, right? Look how big this drop is. That's an NADH drop, right? And then look at the ATP drop, not very big. NADH, really big. NADH, really big. NADH, really big. ATP, not very big. FADH2, really big, but not quite as big as NADH. And again, that goes back to those bonds we talked about. So this might make a little more sense. If you think about bonds being 3 sizes and small bonds liberate a little bit of energy, we capture it as ATP right here. Medium-sized bonds release a medium amount of energy, but we have to capture it. We get 1 shot at it. And so we capture it as an FADH2 molecule. So we don't lose any energy, and then we can exchange that for ATP in the mitochondria in the electron transport chain. And the big bonds are 3 ATPs worth of energy, but we can't capture it as 3 ATP. So we capture it as 1 NADH, so we can capture it once right away, and then convert that to ATP in the mitochondria. All right, so humans are about 38 to 40% efficient in terms of the breakdown of glucose for energy. Before we get into breaking down fats for energy, let's go ahead and start another video because this one's running close to 40 minutes, and it's probably a good time to stop and we can summarize everything with the next one.